Well, good morning, everyone. Welcome back early Saturday. We hope the waffles and chicken, I know they were a new thing to those of you who are not from the South. Certainly was to me before I moved here, but I've grown to like it. Anyway, it's my pleasure and honored to introduce today's moderator for this morning's session. John Charles is somebody that we all know, so I really don't have much to add to what you already know about him, so I'll just kind of go through the basics. John received his PhD from the University of Kentucky in physiology and biophysics, and pretty much from that point on, he devoted his career his professional career here to NASA, here at the Johnson Space Center. I first met John about a little over 20 years ago when I first arrived as a new astronaut candidate, and John was at that time the head of the cardiovascular lab. In fact, on my first mission on STS-65, we flew 2 of his experiments, maybe more, at least 2. We did the collection of the EKG data during entry and landing, and also we had the LBNP, the lower body negative pressure suit, on board doing some operations with that. John, after that, moved on to several other positions of increasing responsibility, including most notably being the mission scientist for the NASA Mir program during Phase 1, as we were preparing to fly our astronauts to the Mir station, and which kicked off, of course, into Phase 2, where we are now with International Space Station construction and operations. He held several other posts, including mission scientist for various flights, and is currently the chief scientist for the Human Research Program, so I can think of no one better to moderate this morning's panel, Life Science Challenges of Long-Duration Human Exploration Flights. So please join me in welcoming Dr. John Charles.
It's always a pleasure to hear oneself described by somebody one admires so much. It always sounds better. I'm very glad to have that happen occasionally. We have an interesting panel in front of us today, and I recall very few instructions, so I'm going to improvise a little bit. We have, by my count, approximately 10 presenters and approximately 8 subjects, which would be approximately 80 presenter-subject combinations in a period of 90 minutes. I think that works out very nicely. Everybody speaks about everything for 1 minute, and then we have 10 minutes for a group discussion. Everybody has a comment for 1 minute afterwards. But failing that, I thought we might try something a little bit different. I didn't want to follow Bill Gerstenmaier's model. I didn't want to copy Bill Gerstenmaier's model because I wanted to be creative and daring. So what I've decided to try to do is I've seeded some of the discussions. I've asked a few people to speak on specific topics, and I've also tried to group some of the topics into groups that, at least to me, make sense. They look like they have some similarities. So if you have your program, you can see the list of topics at the bottom is of course everything we worry about in current space flight, plus a few additional ideas for some perhaps more spontaneous discussion. I've got some categories that I'm going to try and suggest, and that is first, categories related to the duration of exploration class missions. And remember, we're talking about what we call exploration class missions, which is people beyond low Earth orbit. People are always involved in space exploration, whether they're sitting in the control room at JPL or whether they're boots on the ground on the moon. But we're talking about the medical aspects of sending people beyond low Earth orbit. Any place beyond low Earth orbit qualifies for the purposes of this discussion as an exploration class mission. We're looking for long duration human exploration flights beyond low Earth orbit. So topics are generally related to duration, and then to the crew health and well-being aspects, and also related to the novelty topic, which I'm interested in hearing more about, and that is the effects on reproduction and fetal development, which I will tell you frankly is not part of the Human Research Program's agenda. So in the first category, the topics of duration, I think, are related. The topics are aspects of radiation, aspects of food and provisions, aspects of the neurobehavioral, psychological, interpersonal dynamics, and of course the scientific debate regarding the duration of missions. Is more tolerable, is shorter, better, things like that. So to lead off the discussion, I've asked a few of the panelists I've seated with advanced warning that I was going to call on them to speak for just a couple of minutes, 2 or 3 minutes to start the discussion rolling. I'd like to have Jeff Chancellor first weigh in with some thoughts about the space radiation aspects of human long-duration exploration missions.
Okay, Thank you, John. First of all, I want to start off saying it's a honor and a pleasure to be here to discuss this with you. As John said, I'm going to discuss the radiation risk aspect of planning and going on long-duration missions outside of low Earth orbit. But before we talk about where we're going to go, I think we need to make sure we understand how we got there and what has been done in the past. So space radiation research for the space program and for manned spaceflight has evolved with the needs of the mission. During the initial years, we were first trying to just understand and characterize the radiation environment and whether the crew was going to be able to survive or stay sane during the mission until they returned to Earth. But since the Apollo era, the manned spaceflight mission has focused on rapid access to a low Earth orbit that has enjoyed the safe haven of the indigenous shielding provided by the Earth's magnetic field. So a lot of the effort was focused on understanding how the late effects would affect the astronaut crews. Would they develop cancer in their later years once they returned to Earth and retired from the Corps? Would there be effects that were transferred to their children because of the radiation environment in low Earth orbit? So as the focus of the human spaceflight shifts to longer duration missions and the need for crew autonomy becomes much more important and apparent, radiobiology, space physics, and space medicine sciences will need to focus on efforts to develop countermeasures that can mitigate any immediate effects that the crew could incur en route to Mars, or on the surface of any other planet. One thing I want to point out is discussions in the previous panels have highlighted very elegant results of research on behavior or human physiology that had the unique aspect of having results from human subjects to compare to. Unlike these studies, radiobiology research or radiation research cannot use human subjects to look at the very unique space radiation that's found in space. They frequently have to use animal models that have different physiology than humans and frequently have different radiosensitivity and responses. So profiling the dose and the dose rate and the time is difficult for each of these. It varies for each of the end results, and it's even more complicated because the health effects depend on age, sex, the time since exposure, and the particular organ system being studied. Additionally, multiple variables have made results difficult to fully interpret how a dose or dose rate or exposure to this environment that's not found on Earth will induce a similar effect in a human. Some of these, but not limited to, are the radiation responses are not necessarily binary or linear. There are a lot of unknowns in the space radiation environment. I think that's highlighted by the very recent realization that in our own neighborhood, there are 3, not 2, radiation belts surrounding the Earth. The effects that we are looking at are specific to the animal model and focus on a single system response. The models chosen because of similarities to the specific organ systems to a human are sometimes very hypersensitive to the effect being studied. Additionally, technology limits our ability to simulate the space radiation environment. To accelerate a charged particle to energy similar to what would be found by astronauts here on Earth takes machinery that could encompass this entire building. Additionally, since we cannot use humans to do these studies, the computational modeling is quite robust, and it's been till recently that we've had capabilities to realistically model mathematically or computational the radiation effects. So understanding all of the immediate and the late effects of radiation exposure will be critical for long duration missions. A lot of the good work has been done by the space program to understand this, what I would say a very unique stressor that affects every system in the body. But future work should also focus on optimizing what we have learned and minimizing all the uncertainties in order to realistically realize how the health of the astronaut crew specifically will be affected. For the long duration missions, efforts should be targeted towards leveraging new technologies, techniques, and the previous results in order to create more realistic models that account for multi-system responses, the unique dose found by space radiation, the ion species, and energy distributions that will be occurred during interplanetary travel. With that, I will end it and answer any questions. Thank you.
Good, thanks very much, and let's hold applause until the end of the session so we can move on to other speakers, because I think we're going to be hearing from each person multiple times. I'd also like to ask Dr. Vadim Gushin to talk about the psychological challenges of long-duration human exploration missions beyond low Earth orbit and how the research we're doing now may enable those missions.
So, 1 minute for each problem, as it was ordered. I'll try. So, problem number one. The longer you fly, the more is important who is nearby. And that's quite clear from the family issues. To spend with the person a couple of months or a couple of weeks is not the same as having 1 year together. And we see now on ISS that this problem is increasing. That segmentation caused some misunderstanding and wrong perception. That was a question yesterday about that perception. We have a study devoted to the interpersonal perception on the ISS. Now we invited our European colleagues and Canadian colleagues, so that will be a solid study, and we foresee some interesting results, because we do not have that separate house with closed flats in the long duration mission as we had in Sphinx in 1999. That occurred there, and it's famous now that the crew just locked the door between the chambers due to the conflict. 1 minute. Minute number two. But that's not my order.
And you're doing well. Staying on schedule.
Okay. Problem number two is related to performance that we discussed yesterday. We are interested, the crew flying so long, to keep skills. And for a long time, Russia had that famous pilot study that was simulation of docking as virtual reality task made by the astronauts. Now we want to add to this task the virtual model of landing operations and operations on another planet. And moreover, we want to see how the crew can do it immediately after landing. I mean, the first week after landing. And for that case, we not only made a formal proposal, but we built in IBMP the special technological model with a special platform which is moving, and the big screen and the cave and many other virtual facilities. Try to create a system that can allows a sort of diving into the reality, so the crew member can feel that he is really on Mars or on the moon. Problem number three. Communication with the mission control. According not only to Mars 500, but to many examples from the real space flights, the tension between mission control and misunderstanding is increasing. And this is an issue, especially for the delayed communication, for the communication in the extended space flights. And we also launched the proposal to analyze the crew communication with mission control. Of course, only open channel, not the private channel, and of course, only from time to time. And moreover, I want to say that we made sophisticated software, and our British colleagues supposed to make some hardware to analyze the voice frequency, and we expect a lot of results from this particular area. Thank you. 3 minutes.
Excellent. Thanks very much. And I'd like to, I didn't talk to Michelle ahead of time, but Michelle, I'd like you to weigh in on the food and provisions aspects of preparations for long-duration exploration class missions.
Thank you, John. So our biggest challenge is the shelf life of the food. So on a Mars mission, we're going to need a 5-year shelf life of that packaged food system. Right now, our current food system has about 1.5 to 2 years shelf life. So shelf life has several different components. One is it safe? Does it provide the correct nutrition? And is it acceptable? And as the food gets older, the environmental effects are affecting those 3 components. Temperature, the warmer it is, the faster that chemistry is going on in the food. Radiation possibly can help oxidize some of the food components, and those oxidation, some of the food components may be vitamins that are going to degrade. Oxygen provides the oxidation effects, again, losing nutrients, providing rancidity of the higher fatty foods. So what we need to do is determine how do we provide that 5-year shelf life. On the safe side, currently our food is all commercially sterile, meaning that we have provided food that is stable at room temperature. That's fine as long as the package stays intact. And so we'll have to identify packaging materials that won't degrade over time. The nutrition is already a huge challenge. What we're learning is by processing the food to make it safe at room temperature, we are processing it using, typically right now, using heat or other conditions that degrade the nutrition. So how do we find processing conditions such as microwave sterilization, high pressure processing that will not provide as much of a stress to the food and therefore the nutrients won't be degraded? Then over time, the nutrients are degrading again. So can we find environmental effects? Can we find a location in the vehicle that will be colder? Not necessarily frozen, but can it be a little bit colder because that will increase the shelf life significantly. Acceptability is always tied into nutrition. If the nutrition is degrading, so likely is the acceptability. And on a long duration mission, acceptability is that much more important. They are limited to those food items, and if they're not acceptable or they get tired of them, then they're not going to eat as much, and they won't have the right nutritional content in their system to provide them a healthy body. So how do we not only provide the nutrition, but the acceptability so that they will continue to eat, which means we're going to have a lot of food on board, a lot of variety of food, or ways to have them think there's more variety through condiments or other additional kinds of mixes and matches. And I'll leave it with that.
Excellent. Does anybody ever get tired of eating after a long time? I mean, seriously. I never get tired of eating.
So actually, there's some studies that a lot of them aren't published. It's called long-term acceptability. And actually our military has published some work to say, if you eat the same foods over and over, you don't want to eat them anymore. And we've actually seen that pretty consistently with our ISS crew members, that they'll say, at least the freeze-dried foods, after about 3 months of their 6-month mission, they start getting tired of them. And unfortunately, those freeze-dried foods tend to be the vegetables and the fruits, which provide some of the nutrients, and freeze-drying doesn't degrade the nutrition as quickly as our pouch foods do. So, yes, it's an issue.
Good. Thank you. Thank you, Michelle. And I didn't introduce Michelle before. She's has been the head of the food research group at the NASA Johnson Space Center and is now moving into the science management realm. Best luck for that. I'd like, at this point, I'd like to open up the discussion to everybody in the panel. Any additional thoughts you may have about duration-related aspects of long-duration human exploration flights. Duration aspects of long-duration flights sounds kind of redundant, but if you have some additional thoughts spurred by some of the discussion we've had already, now's a good time to weigh in on it. Dr. Pucha, please.
Well, I think whenever we're talking about duration, the big issue is going to be the sustainability of healthcare. I mean, how are you going to do? I mean, it's not going to be like drugs, a bunch of drugs that we'll keep up there and try to use them, but theragnostics, which is now the buzzword for the medical community, we need to be able to diagnose it and use the same kind of a platform to treat it. So theragnostics is going to be one of the aspects I think will be useful for the human exploration, and sustainability of the stability, just like food has. And we have another problem if we just start talking about drugs. It's not only just the palatability but the toxicity. Any compound that degrades, we do not know what kind of byproducts, and so that's going to be a big issue. So I think we need to look into the intelligent medical systems where there is QA, QC, and there is diagnostics, and there is therapeutics, and at the same time there is the cocktail treatments. I mean, we are going to be looking at preventive health more than treatment. That's what should be the motto for pharmaceutics and in the development.
Thank you.
Thank you.
Any additional thoughts on the duration aspects? Mike, maybe having been there for a long time, a couple of times, you have some thoughts about preparations for long-duration human exploration missions.
I'd like to, I'll defer to the rest of the panel on that. The only thing I wanted to really talk about today was radiation, radiation and radiation. So
Please do.
we could go back to that, or we can let you
No,
go
we're
down
still
here.
on, that's still on topic. That's still good.
All right, so can I talk about radiation?
Please do.
All right, because I think I wanted to talk about that. So whether we're talking about this 1 year low Earth orbit missions that we mentioned yesterday in the great panel discussions we had, or if we're going beyond low Earth orbit, to the moon, or an asteroid, or even to Mars, NASA is not, I don't think, speaking from one of the subjects' perspective, is not completely comfortable with how radiation, we don't have a complete good understanding of what radiation does to the human body, and as such, we're keeping things pretty conservative. And so for us to feel more comfortable to broaden the amount of radiation that we're willing to let our American astronauts have, we need to have better understanding and a better understanding of countermeasures. What I'm talking about right now is that the longest duration cumulative time human in space has gone over 804 days in low Earth orbit, and he's the boss at Star City right now. The American limits effectively limit American crew members to somewhere on the order of 500, 550 days, depending on age and gender and things like that. So 550 days dosage of to an American crew member low Earth orbit, that doesn't that means we can't really get to Mars without those current limits. Once we leave the three radiation belts that are protecting our planet, it's going to be different. So we need to understand it, and for us to feel more comfortable so we can get Americans to fly longer duration in space. So right now on board the space station, we have we have a lot of detectors. We have detectors from every country. We even have this big, cool, and we'll talk about it later, the alpha magnetic spectrometer. So I think we understand, there's got to be a huge body of data of what the low Earth orbit environment is. But what is it beyond low Earth orbit? That's a big question. But we spend all our time and efforts on detectors with the tissue equivalent proportional counter, the Canadian experiment, the Russian detectors, the Japanese ones that have great, really nice little radiation area monitors. We carry our own badges around with us. There's about 8 or 10 different detection systems on board, but not one really that I understood after 2 missions and being a commander, a real countermeasure of radiation. So right now our biggest countermeasure is not to fly beyond this 500 and something day. So when I first came here in 2005, after my first mission to the International Space Medicine Summit, we were worried about a lot of things. We were worried about bone mass loss. We were worried about, and we were worried about radiation. The bone mass loss, I think we are less worried about. And this is due partly to the attention that this group has put on it. I'd like to just ask you all to think, and I know it's not everyone's speciality, but radiation's kind of at least from my perspective, from an American astronaut, is the biggest thing I worry about. Not that I'm afraid of radiation, but I'd like to go and like to have some of my colleagues, and heck, these middle schoolers these days, or whoever's going to be going to Mars. I'd like to make sure, just like our Space Administration, NASA, that we're going to send them off, and that we know that they're going to be safe, that we're not going to expose them. So we're going to have good countermeasures in place and an understanding of what radiation is going to do to a human. And to me, that's one of my biggest concerns as an astronaut. So I'd like to ask us to pay a lot more attention to that, because I know the food's going to be fine, and I know we're going to have good exercise, and there's going to be excellent portable tricorders that can analyze our genome and proteins and all that. I feel that's coming. I don't feel as confident about the radiation situation. I know NASA right now isn't fully comfortable in progressing too much farther than we have. Already we're limiting some astronauts we're excluding them from the 1-year mission because they've already had enough time in space, and we don't want to take them over certain limits, and those limits are there for a reason.
Good, thank you. That was very relevant. At this point, I'd like to segue smoothly into an adjoining topic, and that is the topic that I am making of the combined topics on your program of the crew health, conditioning, and well-being, and the omics of human spaceflight. Those factors are related to duration, as are the topics that we've just finished addressing briefly. But I think there's a little bit more of an applied aspect to these kinds of things. Even the omics may have near-term applied aspects that I think we can hear about. So in terms of crew health, conditioning, and well-being, first off, I'd like to ask Dr. Pulaski to say a few words about his thoughts about that topic.
Okay, so again, thanks to the organizers for inviting me today. This is my first one of these, and it's really a pleasure to be here. Some of you know that I've just returned to NASA recently to guide the human research program through the next few years. And our entire focus is on exploratory research, that is research that is aimed at reducing the either likelihood or consequences of risks that we know that humans are being exposed to during long-duration exploration missions. The primary drivers for most of our work are a putative Mars mission that would be in about 2 decades from now, and the long poles in the tent for the Mars mission are radiation, behavioral health and performance, crew autonomous operations, food that we've just heard about, and I would say hypogravity. And I'm going to, because we have just 3 minutes, I'll limit my talk to hypogravity, which is sort of near and dear to my heart. Hypogravity, I would argue that over the last 30 years or so, more money has gone into trying to develop countermeasures to the effects of hypogravity on human systems than any other. Hypogravity affects the bone, muscle, cardiovascular, neurovestibular systems, and has secondary effects on a number of other systems. For some reason, when we started doing human spaceflight, we decided that it was important for us to take along air, water, food, and heat, but we didn't think gravity was necessary to take along with us. As we heard yesterday, maybe gravity is important for us to take along with us. As my good friend and colleague, Professor Young, and partner in crime, I should say, has mentioned in the past, artificial gravity is a topic that's come around and around and around and around. I think we're coming back around to it again. And it's not just artificial gravity to offset the problems associated with zero G during transit. The fact is, we have no idea what 18 months on the Martian surface might do to the physiology of a person. 3/8G is not an area that we can study on Earth. 1/6G is not an area we can study on Earth. We have to have a facility someplace in low Earth orbit to be able to study those things, or we have to look to other species. And I think in the near term, the space biology and ISS National Laboratory folks are going to be designing and flying a rodent centrifuge. And I think if we're smart, we'll make sure that rodent centrifuge is designed in such a way that we can do long-term exposures, a few months at a time, to rodents at, say, Mars gravity, lunar gravity, and begin filling in the curve, at least for bone and muscle, which will translate pretty well, I think, to humans, below 1G and above, below 1G and above 0G, so we can start getting some ideas about that. Other things we can do on the ground, there was, as we heard yesterday, there was an attempt to fly a human centrifuge aboard the space station. It was a trilateral agreement between JAXA, ESA, and the U.S. And unfortunately, the cost of vibration isolation for that system knocked it out of the ballpark, so we weren't able to get it. That would have been a rotating ergometer that we would have been able to get some tests of the efficacy of adding gravity to exercise. Whether that is something that could be reconsidered if the station life were extended beyond 2020 is a question that we'll be looking at to answer in the future, or whether there might be another waypoint in or around the moon, or maybe somewhat more distant, that would allow us to do some human-tended operations where we could build a centrifuge in from the beginning to start looking at these questions as another interesting issue. I think I'll stop there.
Good, thank you. I'd like to segue also into the omics issue and how its relevance to human health and long-duration missions beyond North Orbit. I've got a couple of speakers in mind. First, Dr. Norsk, if you have any thoughts about that topic,
Yes, thank you very much. And I understand I have 2 minutes, right?
more or less.
Yes. Well, since I have so much time, well, just one remark to the food before I start, and that is I totally agree. And if you eat the same every day, you stop eating. I can tell you 41 years ago, my father took me to the United States. We had McDonald's every day for 35 days. And when I came to the United States 2.5 years ago, I had not had one single McDonald's since then. So it really makes an impression on you. But to go back to the omics, well, I, as an element scientist of the human health countermeasures, there's one thing we have to worry very much about, and some people put a lot of emphasis on hypergravity, some on radiation. The problem is the following. Only 24 plus organisms, 24 people have been beyond low Earth orbit, and not very much was done in order to document what happened to the body. And beyond low Earth orbit is not low Earth orbit because you have the combinatorial effects of radiation, of hypergravity, of isolation, of disturbances in circadian rhythms, and so on. And the question is, will that combinatorial effect have an effect on anything or on all things? And there are indications from animal experiments, in particular at Ames Research Center, but also other places, that this is a huge problem. So the bone loss is not just hypergravity, it's if you have a combination of change in radiation and hypergravity, you have an additional synergistic and or additive effect. We need to investigate that. We cannot send people beyond low Earth orbit without knowing that. So we should focus on, when we do beyond low Earth orbit missions, we should focus on the translational effect from the genes over the omics to cellular mechanisms to integrated physiology. As pointed out by Dr. Genges yesterday and Dr. O'Malley yesterday, it is extremely important to connect the phenotype with the molecular mechanisms. So we should think in those terms, and that is what worries us the most. And since I still have 25 seconds, I would like to summarize. 1, ISS during low Earth orbit is not enough to understand what we do to people going beyond low Earth orbit. Even long duration low Earth orbit will never solve that problem. 2, so that's why we have to do it sequentially, of course, in terms of duration. 2, it's the combinatorial effects that are important, not just hypergravity or just radiation or just isolation or just behavioral. And then from cells to integrated physiology, translational physiology, that is the future. And as Dr. Kostovskaya said yesterday, individuals act totally differently in this environment. And even they act differently from mission to mission. So we have to individualize each countermeasure by monitoring each individual and then adjusting this or that countermeasure to each individual. We look different, we are different, and we act differently, and we should consider that more in details in the future. Thank you.
Thank you. And I'd also like to ask Dr. Graham Scott to weigh in on a topic that I think he has had a few thoughts about, the omics issues of people on Earth and especially of people in space. And if you have any favorite foods you'd like to discuss as well.
I actually quite like McDonald's. Dr. Alford, you're not hearing this, right? Thank you, John. And I wanted to acknowledge the leadership of Mr. Abbey and Dr. Alford in orchestrating this outstanding meeting. I wanted to make 3 quick points in my allotted time. The omics, to sort of borrow some language from Dr. O'Malley, is very useful when we don't absolutely know what we're looking for. I don't think we really understand at a molecular level what space radiation is doing to the human body. I think we understand it reasonably well at the level of physiology, but not at a molecular level. Are there particular pathways that are implicated? Are there particular hotspots in the genome? You see, we've made progress in curing some cancers or in doing much better with some cancers because we really understand at a fundamental molecular level what is going on. And we can genotype people, and we can say you have colon cancer and you don't have a mutation in your BRAF or KRAS gene, so you should take Erbitox and it will melt the cancer away. And most of the time, that actually happens. But we don't have that level of knowledge with space radiation. and how it affects the body at a molecular level. We don't really know what we're looking for. Guess what? Despite the heroic efforts of people like Dr. Levine and others to understand the VIP problem, we don't really understand at a fundamental molecular level. We have a lot of theories, we have a lot of great experiments going on, but we don't fundamentally understand what's going on. And I really suggest that, as Dr. O'Malley said, try to collect as much information as possible, and the truth will in some cases reveal itself. I know that sounds like a magic wand sort of solution. It's really not. There are many real examples of that happening. The other point that I want to make is in this area of meta-analysis. We heard a little bit yesterday about databases. So as you collect these huge data sets, for this to be efficient, for this to be really powerful, you have to make this available to the community, because hundreds and then thousands of scientists will start mining it. If it's just this group in this room, or even 50 of these rooms, that's not going to be enough to solve some of these really challenging problems. We need, for the sort of challenges of going to Mars and out beyond low Earth orbit, we need to throw 21st century medicine and all the power of the community at these kinds of problems. So I think the equivalent of a gen bank, which we could call space bank or something like that, is really urgently needed so that scientists can actually start going after this data and making sense of it. And again, to repeat myself, the advantage of the omics is you don't sometimes know what you're looking for, so you have to kind of look at everything to figure out where the needles in the haystacks are. Third and final point, John, in my time. There have been many, many projects. We don't have to reinvent the wheel. The Human Genome Project, the Human Plasma Proteome projects, we learnt a tremendous amount in the course of those projects. I was involved in some of those. We learnt about putting data out quickly. Still, yes, giving scientists the right to publish their data, but putting it out into these databases quickly so that others could see it, so they could mine it, so they could connect up different studies. This is going to be really critical to leverage the lessons we learned about transparency and putting data out quickly and not sitting on data. I want to finish with one real example, and Bill gave me permission to talk briefly about this. Bill has got a beautiful data set of Apollo data, Mercury data, Gemini data, and other data, and he's collated, he's done a beautiful job. That data needs to be put in a database so people can look at it and mine it and make sense of it. And moreover, the specimens that NASA has collected along the way, the blood, the plasma, that needs to be looked at, and that omics data needs to be married with that physiological data so that we can really start to grapple with some of these problems. So thank you, John, for the time this morning.
Good. Thank you very much, Graham. At this point, I'd like to open it up to the whole panel to weigh in on the topic of crew health, conditioning, well-being, and in the traditional sense as well as in that newfangled omics sense. If anybody has additional thoughts on that topic, I'd like to hear about it. Yes, please.
Yeah, I have one comment that is a little bit related to that, and it will only last 15 seconds. There was one thing yesterday I did not understand, and that is people talk about science and operational medicine. I don't understand that. To me, science is not something awkward. Science, omics, basic, fundamental, as talked about by Dr. Scott just before, I think that's just increasing our knowledge base for the operational medicine. So I don't understand this distinction. And then also, for me, science is not an awkward word or some holy things that some people, very clever people, are doing somewhere in a university kind of tower. For me, science just means knowledge you can trust, period. Knowledge you can trust. And will we send people anywhere in space beyond low Earth without knowledge we can trust, without what we do, we trust what we are doing? No. And that's why it has to be a science-based, even fundamentally science-based program, even for operational medicine. There's just one comment, and that's why we have to connect the genes with omics, with phenotypes and integrated physiology.
I hadn't expected such a passionate plea for what seems so obvious, I think, to those of us on the inside. But I think, Dr. Norsky, you're right. If I can weigh in editorially, there's science is just a way of getting the right answer. And if that's a problem, then we're probably not approaching the problem correctly. Any other thoughts on this topic area? Dr. Bogomolov, I haven't asked you to do anything. Have you got any thoughts from your long career and perspective on the duration aspects of spaceflight, or the crew health or omics aspects of spaceflight?
Yes, John. We have a wide panel with many aspects that deserve comments. We have first of all two issues: long duration flights. They are long, and also long duration flights beyond the Earth orbit. Those are two different categories that have to be approached in a specific way. If beyond magnetosphere, we will actually get there and we will send the crew to asteroids or to Mars, we will immediately encounter important problems that are not resolved at this time. First of all, radiational impact and all kind of irradiation in that area. They will have different quality, different quantity in character. At the same time, we will find ourselves in the environment without magnetic conditions, and hypermagnetic conditions will be something that we will not know very well. We are talking about the technical capabilities of creating a special magnetic field and protect a human being, or maybe already now we will need to start research of hypermagnetic conditions and the impact on any living organism. What has to do with the radiation as of today for long duration flights? I am not satisfied at all with the radiational control that we have as of today. We have radiational limits which are established. They are norms based on the practice of manned flights. Those norms are not substantiated scientifically, and they are much greater than what is allowed for nuclear industry workers and submarine workers, such and such. My impression is that the large impacts will be significant. Crew members are probably more resistant. Those are specially selected people. Maybe they will not be bothered as much as the general population. We were talking about omexes. First of all, a human being is a complex, self-regulating system, the system that is able to adjust, to adapt, and use all its reserves and capabilities to be all right in new conditions. And our task is to make sure that they will not be totally adapted to weightlessness, that they will still be able to come back to the ground if we are not leaving them there forever in space, of course. That is why I believe about the radiational impact. We have not resolved issues yet. We have to worry about protecting the crew from radiation, doing that, and also we have to develop new approaches to radiation protection, not only technical by creating thicker shields or special screens or special magnetic fields, but also maybe some kind of pharmacological means can be of protective nature in this case. You remember all our radiational activity research started with developing protection from radiation. When radiation is really powerful, we were working at that time at the military industry, and we were making a bunch of experiments on that. We do not have much research on long-term exposure to radiation. This is the part that is still kind of raw, and it needs to be developed. Secondly, when we are sending people beyond magnetic sphere, we need to make sure that people not only have enough food and water, but they will need to be able to function autonomously, diagnose themselves. They will have to be resourceful enough to make correct decisions in off-nominal situations.
I think that if we're talking about long-duration missions, it means that it's better to have 2 doctors on board at least. Best case scenario, all the crew members will be medical doctors with the necessary level of knowledge base and skill base, because they will be the ones who will be making day-to-day decisions in terms of their nutrition, first aid, and resolve various psychological scenarios or issues that may crop up. Therefore, I think that we are at the very first stages of space exploration in terms of long-duration flights. Now, how can ISS help us in this endeavor? We can go back to radiation protection issues, automation of diagnostic procedures and first aid procedures on board. We can look into medical aid capabilities that would cover not only first aid, but also medical assistance for decompression sickness and life-threatening events that are related to the changes in the environment. So the path forward, or the actions that the crew has to undertake in various scenarios and cases, should be discussed by us further. Some time ago, when we still worked with Mir shuttle programs, we would train the crew when they were already in flight on how they would deal with certain cases and scenarios. And I know that it was very useful. I know we had a lot of training games or simulations that would allow us to identify all of the gaps, all of the imperfections that we have in our system that helps protect crew's well-being. And also I was talking about the fact that human body can adapt to various environments, and we have to make sure that it's done in a smart way. We should have trainers, we should have pharmacological means, and so forth. I apologize, I talked for a very long time. John doesn't know how to stop me, I think. Thank you.
I'd like to thank you very much say thank you very much to all of the folks that contributed to this section. I'd like to move on to perhaps the sherbet, the dessert of the discussion, and that is a topic that's close to everybody's heart, because I'm sure if you've ever spoken in public about spaceflight, you've gotten one and possibly two questions. One is, how do you go to the bathroom in space? Even those of us that don't fly in space get asked that question. And the other, the other concerns, excuse me, the topics of effects of reproduction, effects on reproduction of spaceflight. No kidding, they were listed on the topics for discussion here today. Effects on the reproductive system and the fetal development during spaceflight. I'm not aware off the top of my head if we have any actual experts in that area available to us in the room, but the closest we have is Dr. Chiaki Mukai, who recalls, who, if I understand correctly, recalls having heard once about some fish experiments in space that related to reproduction. I've asked her to sort of discuss this for us a few minutes and see what develops.
Yeah, thank you, John. John always gives me such a tough questions and a tough assignment. And then also and also he gives me only 3 minutes, and I cannot talk faster than you, so it's very hard. But using the 1.5 minutes, I would like to give just a brief summary of what we have learned so far related to the reproduction and also the fetal development issue. It's not the human research program, but the research using animal model. But this is just as far as I know basis. And then the second 1.5 minutes, I would like to list some challenges about this related to this issue. So the first one, talking about the reproduction, actually the Japanese Space Agency conducted the fish experiment, as John mentioned, STS-65 IML-2, which I flew for my first flight. And then according to this, the fish can mate very well in space, microgravity, fertilization, okay, development, okay, and hatching of the baby fish, okay, and then the space-born fish, first generation-wise, it's the rate of abnormality and also the cancer, the same as the one which was born on the ground. So what we know is, as far as the first generation is concerned, it's okay. And then talking about the fetal, fetal, the development issue, there are quite a lot of experiments conducted using, for example, frog eggs, like egg, the development experiment, actually egg is being fertilized before the launch or after the launch, like Ken Souza did it on board, and also newt experiment to understand the otoliths, the development, and seems like the newt developed, they tend to have the bigger otolith when it is actually natured in space. And chicken egg experiment, like USA, like a 2 days young egg and 6 days young egg, and then it was so susceptible to the vibration of the launch. So I don't know if it's microgravity effect on the chicken eggs or not. And the baby rat, STS-90, and then a lot of baby rats died because I think the animal holding facility capability was not well enough to support the animal. So I think, and I think that most of the development issue, the gravity, many researchers think gravity is necessary to support the normal development, especially the neural connections, like neuromuscular development, and like that. And also the coordination. So that is the situation that, as far as I know, I think you folks know more than me. And then based on these experiments that we have conducted using the laboratory animal models on board the space shuttle, and then the challenges is that we really need to know the second and the third transgenerational effect, especially from the viewpoint of radiation effect and also the microgravity effect in terms of the rate of the producing the abnormality and also the cancer rate. That's also the one that we have to do. And the Japanese Space Agency, this is its advertisement, is right now developing the fish holding facility, which can support up to 3 generations the small fish, so that anybody who are interested in utilizing the facility for this purpose, like to see the second and third generation issue, then it's very good. And also this is the benefit of long duration exploration, so that if we have the animal holding facility on board the International Space Station, we will be able to understand, which we haven't be able to done that during the shuttle time. And then the second, so the first one is the challenge is needs to develop that kind of transgenerational facility. And the second is the mammalian is the one that I think that we really need to investigate more to support the human research. So, and so far, I think the mammalian holding facility is not, I think, quite good to hold the animal healthy enough to investigate from the research viewpoint. So we have to develop that kind of issue. So that is the one. And also the third thing, I think that the movement of the research is to understand if the gravity or non-gravity can switch on or off genes. So it's not only for related to the development, but also the regenerative research or regenerative medicine is the one that, like maybe the combined with the iPS cell or something like that, we will be able to open up the new area for utilizing the International Space Station. Like an interesting thing is that we conducted, maybe extra 1 minute if I can get. STS-95 conducted the cucumber experiment, and the researcher find it out some gene is never ever expressed under the gravitational existence, but it's expressed no gravity. So which is negatively controlled by the gravity, that gene. So which is so encouraging because we are living on the ground, which means a lot of potential, the genes are still sleeping on my body. So that if I can go into space and then somehow the switch on my genes, I can be the Superman or Superwoman. So to understand the potential or reserve of the human beings, I think the combination of the omics gene and the gene things, and also the regenerative research with the iPS cell is also the way to understand the regenerative or maybe expanding the reproductive development, or to understand the role, how the gravity plays a role on us. That's what I think. Thank you.
Good. Well, I should have known I couldn't stump Chiaki by asking her for a question that's out of her area of specialty, and you never disappoint, Chiaki. I'm very pleased with that response. Thank you, and you took exactly the right amount of time. I'd like to see if maybe Michelle Perchanik has some thoughts about livestock in space. Is that a possibility for food on these ultra-long duration missions that we might be discussing? I'm trying to stay away from the other obvious implication of fetal development in spaceflight by trying to think of a more utilitarian aspect of it.
Well, livestock, I'm going to say no, only because think about the aromas coming off of the livestock. And so if it's an enclosed environment, I think unless we have an air system to really clean the air, it may not be very practical. In addition, as we continue to hear here on Earth, vegetarianism is certainly the most efficient way to use our food and get our food. And so in a limited environment with limited resources, it just may not make sense to be feeding cattle or even goats or whatever. On the other hand, aquaculture is certainly been considered and should still be considered, because the development of fish is certainly a lot more efficient. It only uses a little bit of nutrients in water, which we always can get, or not easily, but at least have it. some fish even eat waste, and I think some of the studies are showing that the fish are still okay to eat from that, though there's some debate on that. But certainly aquaculture is something that we should be considering for a really good animal protein source.
Good, thank you. Yes, Graham, please.
John, if I could go back to the reproductive topic, just
Please do.
promise to not be too controversial. I just wanted to let everyone know there's actually a series of there's a follow-on to the decadal survey that was published a year or two ago, and Cyril and Mark and Betty Siegel, myself, Dorit Donoval, and one or two others, and actually there are some chairs of the work groups, Dr. Dingus and others in the room. One of these work groups is actually focused on reproduction. I'm actually looking at a outline of their manuscript that they're going to be generating in the next few weeks. And just for everyone's edification, some of the topics they're looking at male reproduction, they're looking at spermatogenesis, sperm motility, they're looking at erectile function, they're also looking at female reproduction, hormone production, sexual function, menopause. So they're looking at these topics, and you'll see this published, in the scientific literature. So I just wanted to let you know that that this is actually being carefully looked at by a panel of scientists that are expert in this area, and so there is actually quite a bit of active work going on. And for anyone who's interested, I have an outline of what the manuscript's going to be. So I just thought I'd share that, John.
Thank you. Yeah, I do wish Dr. Sarah Lynn Mark was here to be part of this panel on this very topic. Incredibly, we are on my schedule, and I would like now to open the discussion to all of the attendees on any of the topics we've discussed. And the topics, again, are on the agenda in your program. Now is a good time to be asking the panelists who are subject matter experts in, as it turns out, every topic on this agenda. Any questions you may have, provide any comments you have, any insights you have, or we can discuss your favorite food items. Yes, please.
Well, I have two very short questions, but as we were finishing up the panel here, which I think is very, very interesting, in my own mind, it was breaking up between two things that we ought to keep in mind when we're going into exploration, as opposed to just living in space. That's one is lifestyle, and the other one is survival. And early on in the program, I think survival is the important thing. Lifestyle will get developed later. Brief question on radiation. Are we only concerned about solar events, or can somebody just mention what kind of the baseline inside and outside of the Earth's magnetic field is? Are we concerned about this long-term exposure without the solar events? And I think we can solve the radiation problem quicker than we can the zero gravity problem. And we all talk about centrifuges, and this gets into the kind of the science fiction area. Has anybody taken a look at artificial gravity, translational artificial gravity, of course, which requires energy, but we need energy for a lot of other things out in space eventually. Has anybody looked at a translational artificial gravity, either speeding up or slowing down?
I can talk to that second one. There have been some unpublished works, to my knowledge, on the linear translation as an artificial gravity, where you would accelerate halfway to Mars and turn around and decelerate the second half. And the engineers that did those analyses at JSC explained to me that the energy cost would be so high that it could probably never be worthwhile, and that rotating the vehicle would be a better way, or rotating something inside of the vehicle.
I can speak a little bit about survival, if you don't
Я
mind.
бы хотел...
Пожалуйста, Валерий Алексеевич, выдай.
Извините, я бы хотел сказать, что вот в вопросах
wanted to say a few words about radiation. When we leave low Earth orbit, we're worried not just about solar events, solar activity. There are a lot of charged particles, heavy particles.
магнитосферы, то она,
And
в принципе,
in terms of magnetic fields,
наших полетах является мощным средством
then I can say
защиты
that
от
it's
различного рода радиационных воздействий.
our protection
Так вот, при
when we do
выходе
orbital flights.
за пределы
Once we
земных орбит
leave near
нужно либо
Earth orbit or low
создавать
Earth orbit,
искусственные эти магнитные поля, чтобы обеспечить часть
should
защиты
we
от радиационных
create
воздействий, с одной
artificial
стороны. А с другой
magnetic
стороны,
fields to ensure that we still have some level of protection? Or
с гипомагнитными
should we
условиями, если такого магнитного
do more
воздействия
research into hypomagnetic environments if we are to do without magnetic fields altogether? These things could be pursued in parallel. I know that both
разработки, изучения
fields are
влияния, а также
in the very early stages of research,
отрицательных эффектов. Это что касается
but it's something that we need to pursue further. That's what I had to say about radiation. I apologize for the interruption.
I'd like to speak about survival and autonomy. Of course, radiation is very important. But I think that for the most part of the audience, autonomy is just the opportunity to do what you want. And that's, of course, better than to follow the strict recommendations of the mission control. But when we discuss food, for example, and nutrition, we just think that it's like McDonald's nearby, and we can just order fish or meat, and it's so easy. Each gram will be expensive and precious. And that's, from the psychological point, it's real survival. If you just do not dislike, but you have no food that you really need, that's what we have to discuss. And, for example, imagine what happened in one of our simulations in Moscow. When the crew, they just preferred one type of food and disliked another one. So in the middle of the mission, they just didn't have the food they like. So we have to speak about this, not about the preferences, but about the shortages and what kind of conflicts could appear in the crew when you cannot resupply the crew. Just autonomy is not that kind of is not that kind of nice thing from that point. And decision making is not that easy if you have to decide every time, not just in the situations you'd like to decide, but in the situations when it's high responsibility, risk, and the crew is divided. When there are aliens in the crew, when it's subgrouping in the crew, how to manage the crew under that autonomy. And finally, to sum up, I want to pinpoint the message from Professor Bogomolov that we are not doing the proper training. Even that games on the ISS could give us a lot of information about autonomy, about decision making, about resources distribution, about the crew behavior, about the interaction with delay, and many other issues. And finally, if the crew is having enough data to decide, yesterday Professor Bogomolov told you that we had to bring crew members back because of the lack of data. But in the autonomy, that's the crew members. They have to decide if they have to use some medical drugs or medical care, and we are not yet providing them enough data to make decisions. So our logistics aboard is for us, for mission controllers, for the scientists, but not for the crew. Think about that from the point of view of the survival.
Thank you. And I think we have a comment or question from Dr. Kerwin.
A question for Dr. Chancellor. Since I believe that the only way we're going to solve the radiation problem, that is determine what the real effect of cosmic rays is to go fly. When we start flying in deep space, can you mount a research program which will effectively reduce the uncertainty about the relative biological effectiveness of cosmic rays?
That's a good question. It's a difficult question. In in response to that and a lot of the discussions about radiation, I think sometimes the problem is oversimplified, and that is a fault of possibly the community and our failure to communicate how difficult the problem is to people who don't do the research itself. And that is really because we don't understand it very well either. Everything that we measure is a moving target, literally. The metric we use to compare and quantify the exposure changes as it moves through tissue. So it can be done. It'll be difficult. But one thing I maybe didn't emphasize very well initially is the research community responds to funding. And if the program directs them to look at specific areas, they will. And it's not, and we don't understand it because there isn't a large passion in the area. It's just because it's a very difficult and multifaceted area to look at. And I'd like to say one thing off the top of my head. The idea of a magnetic shield is very novel and sounds kind of cool, but just very quickly, with a very partially caffeinated brain, I think that it could also add more difficulty because now you're creating drag because you have a magnetic field generated around the aircraft. Space and volume and mass is already very, very valuable. And so now you have to create a mechanism, machinery, and an energy source to provide the magnetic field too. So it could solve the problem for radiation, but it may also create many more problems that are not easily overcome.
I'd like to assert moderator's privilege by asking if Dr. Sam Ting has some thoughts about the radiation aspects of deep space flight and long-duration human missions. And we have a microphone coming down. And I will say before the microphone gets to him that I'm reminded by the presence of Dr. Kerwin that so often we have put people, astronauts, into situations and then the science has caught up with them, as happened on Mercury, Gemini, and Apollo missions with progressive increases in duration. And I won't be surprised if that's exactly what happens. The case you suggested to Dr. Chancellor, where radiation monitoring occurred in parallel with long-duration missions beyond Earth orbit, is the way that we acquire information for subsequent crew members' safety and health and protection. Okay.
Just wait a second, I'll show the slide.
Good. Can I have one other question, then perhaps Jeff Hoffman has something to say real fast?
Just a quick one. This is for Michelle Perchonok about food, and I'm curious what some of my astronaut colleagues would think of this. I've always thought that it would be wonderful, both from a crew morale point of view, but also from a nutritional point of view, if we had the ability to bake bread in space. And I'm actually serious about that for two reasons, because I suspect that it's easier to keep raw flour and the other nutritional ingredients, healthy for extended periods than actually, once the bread is baked, it goes stale. So is that a reasonable thing to think about?
So yes, it is. And in fact, because we had limited time, I didn't even want to go in that direction. But there is going to be, we've started the research on sort of trade study level of does it save mass? What happens to the nutrition, et cetera. But yes, a bioregenerative food system of some sort, I would say, is inevitable. Now, likely what will happen, I'm going to go really quickly on this, is we'll grow fruits and vegetables. That will give us the nutrition as well as bright colors, wonderful aromas, crunchy textures for that acceptability.
We would love to garden. We would love to garden in space.
Yes, and
it
not
would
only
be psychologically as well as nutritionally great.
So yes, there's a psychological benefit to tending the garden as well as the results. What we're proposing right now, and we're at really early stages, is bringing up in bulk wheat berries, soybeans, peanuts, other dried beans, and using those to mix and match and make items. And then the question becomes, what would be the most important item to do? And though we haven't done the study, just gut feel, baking bread is going to be one of those first things. Maybe baking cookies or something like that would be, and then eventually making pastas, cereals, tofu, other items like that. But yes, that is a whole other piece and won't really happen except for the garden piece until we're on a surface mission for long periods of time, because there is that volume mass upcharge in the beginning for all the equipment. But I feel very strongly that we're going to be doing some of that on board a long-term mission. Oh, right. So Bill reminds me that 3D printing, and we've actually got a Small Business Innovative Research Grant, the Phase 1, starting up hopefully in a couple months. Being able to say we've got this protein, carbohydrate, fat paste, and that may not be the right texture, but you add in nutrients and flavors so that now you have a very nutritious, whether it's a cookie or a pasta or something that provides you with unique products at a very, we think, could be a very economical on mass and volume, because now you're picking and choosing flavors and nutrients and then mixing and matching up there. So again, in variety. So we're looking at all that. It's might be a hard sell in the long run, but I think for long-term missions, we're going to have to do that.
Very good. And Dr. Ting, if you have some thoughts about the radiation questions.
Yes. For the AMS experiment on the International Space Station, we have spent about 10 years to build a superconducting magnet. So I know a little bit about the issue of building a superconducting magnet in space. Superconducting magnet is very useful for cosmic radiation, for propulsion system, for energy storage, and energy generation. Could I have the next? So, to my understanding, a trip to Mars and back takes 3 years. Cosmic radiation, once you leave the Earth's atmosphere, Earth's magnetic field, is 90 rand per year. Lethal dose is about 300 rand. So if you don't have protection, perhaps there's no need to come back. And for radiation protection, if you build a superconducting magnet, it's about 30 tons. If you use material, it's about 1,000 tons. Go back. Go back.
We've got a big rocket coming.
I've studied this at quite some length. A superconducting magnet, there are 2 coils. Superconducting coils has the property, once you charge it, you will last for 5 or 6 years because there's no resistance in the coil. And if you arrange the coil in this way, there's no field outside, no field inside. So the astronaut can go out, the astronaut can stay in the crew compartment. Next one. So the top left picture shows that even though the cosmic radiation is 90 rand per year, and the lethal dose is 300 rand, on Earth, we have Earth's magnetic field is 500,000 Gauss meters. And we have 10 kilometers of air, which is equivalent to 10 feet of aluminum. This is why we're here today. And also you are protected by Earth's shadow. On the space station, the Earth's magnetic field is 360,000 rand. And therefore the radiation is 20 rands. That's why astronauts face. If you want to go to Mars, if you use material, you need 1,000 tons of aluminum or 500 tons of methane. Then the radiation is about 20. To get equivalent 20, and you need 30 tons of magnet. Next one, please. So, biological measurement, I am not qualified to say but as an experimental physicist, physicist, I will ask myself, how are you going to do tests, which people already mentioned. For passive shielding, the question is to develop a new material. New material, it's quite difficult because we know everything on Earth are made out of protons and neutrons. And you're not going to generate a new material. Superconducting magnet, the technology exists. To do it is quite difficult. Took us nearly 10 years to build this. Next one. So we have done a quite careful engineering study, how a magnet will look like, and with the 6.2 Tesla magnet around the crew compartment and around the front, including the thermal shield, including the supporting structure, and including superfluid helium, is total 30 tons. This is based on current technology. But the key point is, once you charge it, you don't have to recharge again, because the coil has no resistance. Next one. So this is the only way. If you go to Mars, you can come back. This is how I understand it. Thank you.
Good. I can think of no better way to conclude our session this morning than with this presentation and this cartoon that we just saw. So in my duty as moderator to summarize, I will tell you the things that I'm taking away are that the radiation countermeasure, notwithstanding the last option that we've just discussed, is still the outstanding problem, and that needs to be resolved. One of the things we need to think about, in addition to radiation, is the absence of gravity and its long-term effects, both at the genetic level and at the macro level. I think we heard a couple of times that if we want to get smart about how to survive on long-duration flights beyond low Earth orbit, we need to do long-duration flights beyond low Earth orbit, perhaps invoke the pioneer spirit that got us this far, and accept the level of risk, the higher level of risk than perhaps we're comfortable doing right now, to learn exactly what the problems are. I've also learned more than I thought I would about the possibilities of reproduction and fetal development in spaceflight. And I am piqued by the notion that food seems to have run through the discussion this morning, culminating, or at least with the penultimate comment, of the possibility of actually printing 3D food in space. So I want to thank the panel members for being available to us today. I want to thank the audience for your questions, especially Dr. Ting for his insightful comments at the very end. And I believe now is time for a break to continue the discussions offline. So thank you very much for being here bright and early on a Saturday morning.